The present demonstration shows the integration of existing data sources towards a global surveillance network for molecular epidemiology, based on service-oriented architecture and grid technologies. Its relevance is being tested through the current H1N1 outbreak. Recent years have seen the emergence of diseases, which have spread very quickly around the world through human travel and animal migration. In late March 2009, cases of human infection with H1N1 virus were first reported. At the time, more than 100 countries had reported cases of H1N1, and WHO has officially classified this outbreak a pandemic in response to its worldwide geographic spread. In the framework of the current project, we are wondering if there is a way to improve the response to pandemic using grid. In order to determine the spatial and temporal dynamics of influenza A virus during current pandemic, molecular epidemiology will permit us to track down the virus history through its imprint on the viral genome, and thus provide epidemiologists with elements for their decision. There has been a worldwide effort to isolate and sequence flu virus genomes. From test specimens from patients or animals presenting clinical or epidemiological evidence, specific subtype identification is achieved. Specimens with a positive result are sequenced and deposited in influenza databases. Every day the NCBI-FDP server is updated with new sequences of H1N1 segments, with 7 files, sequences of nucleotide, protein, and coding region, and corresponding metadata. A grid database is populated with such data through an automatic synchronization. The idea is to federate several virus data sources, including biology and veterinary research institutes, in a common database daily updated. Then these data will be processed on grid resources. Through a web interface, users will be able to visualize dynamical results. The current prototype starts with a sequence preparation in correct format, then a multiple alignment using muscle, followed by a curation with G blocks to identify conserved blocks. From this step, a phylogenetic analysis is performed to obtain a branching diagram. The tree-in software permits the visualization of a phylogenetic tree annotated with latest sequences highlighted. The current prototype is called GINFO, Grid-Based International Network for Flu Observation, and is available in the corresponding website. In the result web page, one can select a date as well as different branching diagram. An annotated tree with last sequences colored is obtained.